Today I'm going to talk to you guys about types of fish migration, and we're going to focus on differential migration. Some common terms that we're going to encounter today are migration, and that is defined as direction of movement of an organism between specific areas. I'm sure you guys have heard of this term plenty of times before. We're also going to talk about adopted migration, which is movement acquired from social learned behaviors. These experiences are often learned by parents and passed on to juveniles and have been being done for generations upon generations. They are transmitted through interactions or numerical dominance. Interpretive migration is the expansion of a group's abundance and spatial distribution, often associated with mass movements by a population into a novel habitat or ecosystem. Partial migration is the coexistence of two or more life cycles within the same population, traditionally an individual of a migratory or sedentary lifestyle. Partial migration has three dominant types. There are the non-natal divergence, the natal divergence, and differential migration. Non-natal divergent is when one individual stays in the natal habitat, then others disembark but return to spawn. This is a classic type of partial migration. Natal divergence is when residents and migratory individuals commingle during non-breeding season. They originate from separate natal habitats and are termed breeding partial migration. Differential migration. This is what we're going to be focusing on today. So basically, it's migration that is dependent on size, age, sex, and other attributes. Size and sex can lead to other types of partial migration, such as natal divergence, skipped spawning, and straying. Switching is a midlife abrupt change in migratory behavior. So with this figure here, we're going to look at the why, when, where, how, and what factors play into fish migration. As you can see, the external factors of why and when are focused on the food web, which is the chances of foraging, and population density and predation rates, and abiotic conditions such as changes in the seasons. For internal state, for the why and when, we're going to look at things such as size, energy storage, physiology such as neurohormonal status. These all will determine the individual's readiness to move. And we also have to look at the where and how of the organizational design of the individual. So they're going to have things like sensory cues, inherent imprinting, learning, and size and morphology are all going to play a role in the individual's ability to migrate. We also want to look at the path of movement, which is the what, and this can alter the individual's environment and internal state, and it can influence an individual's decision to continue moving or undertake a sedentary lifestyle. And these are just movement phases, and they can be tracked throughout an individual's lifetime. This figure shows factors for fish migration that are related to the cost, energetic costs for an individual to move based on size. As you notice, this is on a logarithmic scale, and as an individual becomes larger in size, it becomes easier for them to move through the water column and migrate further distances. There's another thing to note is that birds have a greater travel cost than marine mammals do because they have to focus on both the uplift, which marine organisms don't have to focus on because they are in a suspended water environment and often have water buoyancy controls such as a swim bladder or a fatty liver. Fish locomotion can be affected by a multitude of different things. Their caudal fin is a big factor on their speed at which they swim and their maneuverability. So fish with a more forked tail are going to be a lot better at cruising, while fish with a more rounded tail are going to have a lot more maneuverability. Fishes that are better at cruising with a forked tail, you can think things like a bluefin tuna, and then something that's more maneuverable, you can think something like an eel. For the paired fins, it's going to have a factor of undulation versus oscillation, and this is going to affect their ability to maneuver as well. Other factors are going to be the lateral recoil, so that's how much they almost move back and forth as they swim through the water column. So these are all going to be factors in their ability to feed, evade predation, reproduce, and migrate. There are lots of different types of technology used to help track migration. As you can see in the figure, the different types of technology play a big factor in how long they can be used for and the spatial scale that they can be used for. There are a lot of different costs and benefits for doing these different types of migratory tracking methods. For example, visual radar and sonar snapshots are very short time frame, and you can't track for such a long period of time, but they're very cost effective because you can just get on a boat and take pictures of a shark. An example of this is this image right here. A scientist or a team of scientists could take a picture of a shark fin and see the indentations and how the fin has unique features, and they can use that as a way to identify that individual and see how often it is coming back to the area that they are in. Some acoustic telemetry is a system that is used by planting a acoustic tag either internally in an organism or onto its dorsal fin, and it will have a hydrophone receiver that detects that individual's unique acoustic tag and can tell people where that fish is migrating to and what time of the year that fish is in that location at. Radio telemetry is used with an antenna and a receiver. Also, something that's used pretty often is GPS satellites. Those are great for tracking large scale things, but they do not get as fine of detail as you're going to get when you do acoustic tagging or photo tagging tracking. This is a video of a shark being tagged with an acoustic tag.
The shark is named Catherine by cat customers in honor of Catherine Lee Bates.
We're taking a stroll at the Sebastian Inlet Park, about 140 miles north of Miami. Researchers tagged this 2,300-pound great white shark all the way up in Cape Cod, Massachusetts back in August. They spent about 15 minutes with her. They did an ultrasound. They measured her. They even got a blood sample. And they put this special tracking device so they can keep tabs on her, not just for right now, but also for the next 5 years. So far, this shark has traveled more than 36 miles. And the reason they're doing this is because they're trying to unravel the mystery behind the great white shark in the Atlantic Ocean. They want to figure out where and when these sharks are breeding, and also where their nurseries are located so they can protect these areas. Now, the cool thing about all of this for you and I is that we can actually keep tabs on this research real time, and we're all waiting to see where Catherine will head next.
It is so important that we understand where sharks are going and where other marine organisms are going so we can understand their breeding grounds and their mating grounds, and we can help to protect and conserve many of our endangered species. So differential migration is migration within a population dependent on size, age, sex, and other attributes. Size and sex can lead to other types of partial migration, such as natal divergence, skip spawning, and straying. Switching, which is midlife abrupt shifts in migratory behavior, is a type of differential migration. So for natal divergence, the individuals were originally from separate natal habitats, and the residents and the migratory individuals will commingle during non-breeding season. This is termed breeding partial migration. Skip spawning is something that occurs when all the fish of a species disembark to the natal habitat as a mature adult. Only a portion of the adults will take this natal migration every year. An example of this would be a surgeonfish. They begin their life as an egg in a major river, and as they hatch into a juvenile, they spend their time in these rivers and then in deltas. As the juvenile grows, they migrate to an estuary, and they continue to grow and mature until about 15 years of age, and then at that time they will decide to go back to the river that they have been imprinted upon and lay their eggs for the next generation to come. Non-annual and delayed spawning are believed to be pretty common in marine fish, and especially in species that live for a long period of time. Straying. This is when migratory individuals reproduce in non-natal habitats. Straying tendencies often vary by size and sex. This is considered an amended type of non-natal divergence. As we remember from before, non-natal divergence is resident and migratory individuals commingling during non-breeding season. An example of this is in the New England Channel when placids, which is a type of flatfish, spawn flows to various nursery habitats. Size migration is different spatial behaviors over the lifespan instead of having a single behavior throughout the whole entire lifespan. Changes occur most rapidly in the larval and juvenile periods because of a change in swimming performance, foraging success, and predation chances. Fish leave the natal food web and enter a new one based on size. You can think about this as a planktonic larva is going to forage on the planktonic level, and they are going to eat things such as zooplankton, while once they leave that plankton larval stage and become a juvenile and an adult, they are going to begin to feed in the nekton. So this is the figure that we saw earlier, and as we remember, this is on a logarithmic scale, and as a fish increases in size, it's going to have a lower cost of energy to swim. So this means that larger fish are going to have a larger range of area that they can occupy and migrate to and from within the oceans. Shellfishes such as cod, flatfish, snappers, and drums are going to have a wide range, as well as coastal sharks such as lemon sharks, bull sharks, and angel sharks, compared to some of our smaller fish like our killifish and our mummy jugs. Some fish have longer migrations in juvenile stages than they do in their adult stages of life. Examples of fish that do this are the Atlantic and Pacific bluefin tuna, as well as the tiger and white shark. Atlantic and Pacific bluefin tuna have transatlantic migrations more frequently as juveniles than as adults. And for the tiger and white shark, larger migration ranges are found in the juveniles because they are exploring, foraging, and avoiding cannibalism. Size and sex play a big interaction in differential migration. Sexual dimorphism is very common in fishes. Broadcast spawning and parental care both occur, causing variation in size and survivalship. There are many examples of sexual dimorphism in marine fishes. One example is the striped bass. The female striped bass matures later than the male striped bass. They grow larger in size and have a larger migration range. Female fertility success is more dependent on size than it is for males. Another example of sexual dimorphism causing differential migration is the scalloped hammerhead shark. The females migrate farther off the shore to find more energy-rich pelagic prey. This causes them to grow faster and larger than the males. After maturing, they migrate to support gestation of their large offspring. Opposite-sex bias occurs in connected marine and freshwater habitats. Examples of this are the Australian barramundi, where the males invade the freshwater habitat. And another example of that is the tupung, which is a type of Australian ice fish, and the female will be seasonally found in rivers. For shark courtship, females often receive a bite wound. This leads to segregation of the sexes in differential migration. As we remember from before, differential migration is migration within a population dependent on size, age, sex, or other attributes. So this is a sex-related factor of differential migration. Examples of sharks who experience this are going to be your mako sharks, your white sharks, cat sharks, and spiny dogfish. Live birthing sharks have the cost of pup bearing, and this leads to them having a more limited range of habitat. This is also a type of differential migration. Resource competition and territories can also play a factor in differential migration. For cardinal fish, females control the spawning habitat, and winter migrations lead to reacquiring spawning territories. For salmon species, they never lose their spawning territories. And then for midshipmen, they have size-dependent territories, and body size dependent territories. So the time that a midshipman fish can remain a resident of an area is dependent on the size they are as a fish. And this is my works cited page.